I became interested in animal ethics in an undergraduate philosophy class, and I'm committed to living these values from my diet to my work in lab. As a scientist, I chose to work in this area because I believe it's one where technological progress can make a real difference, unlike other issues where education, political advocacy, and personal lifestyle changes are needed. My aim is to contribute to this progress by developing more faithful models of the distinctive human brain, so that researchers can apply them to study complex neurodevelopmental processes and diseases in a more realistic context. I'm working with brain organoids, which are tiny, simplified tissue culture models grown from human stem cells, and testing ways of enhancing these models by adding a source of internal fluid pressure, which is present in the real embryonic brain, and helps guide its organization as it grows. Because these in vitro tissue models grow in trays and dishes, where they're directly accessible, I can easily perform hands-on experimental manipulations as they develop over time. Thanks to this accessibility, I can explore how mechanical forces influence human brain tissue development, using cell-based models with human genetics, regulatory mechanisms, and developmental timing that can give us a closer look at time points when we could never have this kind of experimental access to the real developing human brain. I joined Paula Arlada's lab to study developmental neurobiology and advance in vitro models like these with some of the world's best brain organoid researchers. Her experience in mammalian brain development and devising ways to model it in a dish have been essential for my progress in the project, as has the help of my many lab mates and collaborators. So far, my preliminary experiments have provided evidence that adding pressure to brain organoids not only extends the time span during which their overall organization resembles that of the real developing brain, but unexpectedly also appears to regulate neuron development. With funding from IFER, I'm using single-cell analysis techniques to take a closer look at this effect. I'm also testing ways to make this technique work at a larger scale and with less manual input to potentially facilitate its wider adoption as a platform for in vitro brain development research. I'm especially excited to use IFER's fellowship grant to test new synthetic alternatives to the animal-derived reagents included in the protocol I adapted for this project, so that if this approach is adopted more widely, it can be in a form that's more cost-efficient, consistently reproducible, and humane. I'm grateful to IFER for its support as I complete this project. After I graduate, I'll continue relying on my experience from the work I'm doing as an IFER fellow as I continue to work with similar human cell-based organ models, study how they can be applied in higher throughput and commercial contexts, and advocate for regulatory and funding policies that help promote these new tools as part of a more patient-relevant, reproducible, and humane research ecosystem.